Good afternoon. It is the presentation for pre-PhD coursework seminar. My research topic is characterization of Escherichia coli isolated from Bagmati River water, Kathmandu, Nepal. I have enrolled PhD course in Dr. Babasaheb Ambedkar Marathwada University, Sambhaji Nagar, Maharashtra, India, in the year 2019, and my research supervisor is respected Sir Professor Dr. Rajendra D. Joshi, Yogeshwari Mahavidyalaya, Ambajogai, Bid District, Maharashtra, India. Introduction. The Bagmati River, which flows through the heart of Kathmandu, serves as a primary water sources for various purposes like domestic, agricultural, and industrial purpose in the Kathmandu Valley. It is the holy river of the Hindu and Buddhist communities. Along the river banks, one can find a multitude of significant holy sites such as Gokarneshwar, Pashupatinath, Guyeswari, Tekudoban, etc. Over the years, the river has experienced severe degradation due to the discharge of untreated sewage, industrial effluents, and other solid waste to the river. There is an urgent need to assess the pollution level of the river and characterize the extent of fecal contamination, particularly through the study of Ecoli in the Bagmati River water. The river's water sustain a diverse ecosystem supporting the myriad of plant and animal species along its banks. The fertile lands adjacent to the river banks benefits from the nutrient-rich sediments deposited during the river's course, fostering agricultural productivity and contributing to the region's abundance. The Bagmati River, which flows through the study will investigate the prevalence, genetic diversity, and antibiotic resistance pattern of E. coli in the Bagmati River, providing valuable information about the water quality of the river. In addition, this study will contribute to the growing body of knowledge on E. coli in riverine system and help inform remediation strategies to protect and improve the microbiological quality of the Bagmati River water. The main goal of this study is to comprehensively characterize Escherichia coli isolated from the Bagmati River water in Kathmandu, Nepal, to assess the level of fecal contamination and potential public health risks associated with it. Objective. The general objective of this research is to characterize the E. coli isolated from the Bagmati River water in Kathmandu, Nepal. And there are several specific objectives of this study, which are as follows: to assess the different physiochemical parameters of the water, like temperature, pH, DO, BOD, COD, nitrate, nitrite, nitrogen, phosphorus, ammonia, and heavy metals ion concentration, to isolate and identify E. coli strain from water sample, to assess the phenotypic characteristics, including virulent factor of E. coli isolates, to determine the antibiotic susceptibility profile of isolated E. coli strain, and to analyze the genetic diversity of E. coli isolates using molecular techniques. Materials and methods. Study area. Bagmati River of Kathmandu, Nepal is the study area of this research work. Sampling site and collection of sample. 7 major points were selected for the collection of water sample from the Bagmati River, which are as follows. Sundari Jal, the uppermost portion of the river just outside the Shivapuri National Park, which is considered as the minimum human influence area. Next, Gokarna, Guyeswari, Subidanagar, Sankhamul, Teku, and Chobar which is the exit point of the Bagmati River from the Kathmandu Valley. This is the map showing the Kathmandu Valley. The Bagmati River flew through the Kathmandu Valley, and the red dots in this map shows the sampling site of the research. Analysis of the samples. The present investigation studies on characterization of Ecoli isolated from Bagmati River water, Kathmandu, Nepal. The study was carried out in Department of Microbiology, Yogeshwari Mahavidyalaya, Ambejogai, Bid District, Maharashtra, India, during the year of 2020-2022. Physiochemical and bacteriological analysis of sample. Physiochemical analysis of water sample as temperature, pH, TSS, DO, BOD, COD, total nitrogen, phosphorus, nitrate, and metals like copper, lead, zinc, cadmium, chromium, mercury, and total coliform count were analyzed by standard method mentioned in APHA 2005 guidelines, that is 23rd edition. Samples were collected from the different sites of the selected area in white mouth polythene bottle and stored in icebox. After collection, a portion of each sample was immediately transferred to the laboratory and stored at 4 degrees Celsius for microbial analysis. Physiochemical parameters of the sample were analyzed as the methods recommended by APHA 2005. Isolation and enumeration of bacteria was done by MPN, that is most probable number, and power plate techniques. The isolates were identified by the study of cultural characteristics, that is colony characteristics, gram reaction, and biochemical properties such as Imbic test, TSI reaction, urease test, sugar fermentation, etc. by following the standard microbiological techniques. Species level identification. For species level identification, genomic DNA of potent bacterial isolate was extracted by a standard protocol and analyzed on 0.8% agarose electrophoresis. Further PCR was carried out to amplify the 16S rRNA gene of the extracted genome DNA of bacteria using gene AMP PCR with forward and reverse primer. The obtained 16S rRNA gene sequence were assembled and exploited for polygenetic analysis. The 16S rRNA gene sequence related taxa were acquired from the Gene Bank database at the National Center for Biotechnology Informations. The report, which are sent through the online, includes NCBI blast, and all the results are found in the online given here. This is the photographs of water sample collection in the sampling site of Bagmati River. Flowchart for the methodology. First of all, water sample were collected from various sites of the Bagmati River in Kathmandu. Physicochemical and heavy metal analysis of the water sample were carried out by standard protocol, by following the standard protocol. Evaluation of total coliform bacteria and isolation and identification of Escherichia coli were done. Antibiotic resistance pattern of E. coli against various antibiotics, various common antibiotics were performed. And confirmation of E. coli using 16S RNA gene sequencing and phylogenetic analysis were performed, and then the result were interpreted. Results. Altogether, total 28 water samples were processed throughout the study period of 2 years in each season, dry and rainy, of the years 2020 and 2021. 7 water samples were collected from different sampling sites along the river. All 7 water samples collected from January to June 2020 shows temperature and pH level within the range of WHO standard. However, other physiochemical parameters were above the permissible limits of WHO guideline, except the sample W1 that is collected from the Sundarizal. The results are tabulated in table number 1. The heavy metal ion concentration of all samples were within the WHO standard, but the mercury concentration in sample W7, which is collected from the Chovar area, exceeded the permissible level of the WHO standard, and this result is also tabulated in the table number 1 in next page. The bacterial load in all water samples exceeded the range of WHO guideline. The results are presented in table number 2. E. coli was the predominant bacterium among the total isolates. This is the table number 1 that shows the physiochemical parameters of water sample collected during January to June 2020, which is already mentioned. And this is table number 2, shows the total coliform count of the water sample collected during January to June 2020. And the sample collected from the Teku site shows the higher load of the coliform count, that is 148.6 multiplied by 10 to the power 2 CFU per ml. Results of water sample collected in July to December 2020 is shown in table 3. Physiochemical parameters of the water sample were exceeded than the permissible level of the WHO guideline, except the sample number S1 that is collected from Sundarizal. Heavy metal ion concentration of all samples S1 to S7 were within the range of WHO standard. The results is tabulated in table 3. Bacterial load of all water sample was exceeded the range of the WHO guideline. The results is tabulated in table 4 in the next page. E. coli was the predominant bacteria among the total isolates. And this is table number 3, which shows the physiochemical parameters of the water sample collected during the period of July to December 2020. And this table number 4 shows the total coliform count of the water sample collected from Bagmati River during the period of July to December 2020. Water sample collected from the Bagmati River during January to June 2021 also shows the same results as results obtained in the same duration in 2020. That is, all parameters of the physiochemical test exceeded the WHO standard, excluding W8 water sample, which is collected from the Sundarizal sampling site, and the result is presented in table number 5. Concentration of metal ions of all water samples, that is W8 to W14, were within the range, within the limit of WHO standard, and the results are presented in the table number 5. Bacteriological analysis shows that all tested water sample have high range of bacterial count than the WHO guideline. The table is the results is tabulated in table number 6. E. coli was the predominant bacteria among the total isolates. This table number 5 shows the results of the physiochemical parameters of the water sample analyzed during the January to June 2021. This table number 6 shows the results of the total coliform count of the water sample collected during January to June 2021. Physiochemical parameters of the water sample of Bagmati River in July to December 2021 were beyond the range of the permissible level of WHO guideline, excluding sample S8, which was collected from the Sundarizal sampling site, and results are tabulated in table number 7. Heavy metals ions concentration of all water samples were within the range of WHO guideline. Bacterial count was higher than the WHO standard. E. coli was the predominant bacteria among the total isolates, and total 28 E. coli isolates were selected for further study. And this is table number 7, represents the physiochemical parameters of the water sample analyzed during the period of July to December 2021. And this table number 8 shows the results of total coliform count of water sample of Bagmati River during the period of July to December 2021. EST pattern of the E. coli isolates. All the isolates were resistant against the antibiotic ampicillin and erythromycin, but all the isolates were susceptible towards the antibiotics like chloramphenicol, nitrofurantoin, and genthamycin. The results are tabulated in table number 9. This is the photographs of AST pattern of the E. coli isolates. The isolates were identified based on the morphological characteristics, size, color, and texture, and microscopic examination. On biochemical characterizations, the isolates were identified as E. coli and were confirmed by 16 years rRNA sequencing. On blast analysis, the multiple sequences alignment of the isolates shows 99% sequence homology to E. coli. And this is the phylogenetic tree E. coli. And conclusion. In conclusion, the comprehensive assessment of the water quality of the Bagmati River in Kathmandu, Nepal, over the period of January to June and July to December for the years 2020 and 2021 revealed significant insight into the environmental health of these crucial water resources. The key findings and implications drawn from this study are outlined below. Physiochemical parameters of water. The physiochemical analysis of water sample indicated that while temperature and pH generally meet Nepal and WHO standard, several samples exhibited elevated level of the total suspended solid, dissolved oxygen, biological oxygen demand, and chemical oxygen demand, surpassing the recommended guideline. Heavy metals tolerance. The study highlighted that all the isolates from the Bagmati River exhibited a remarkable ability to tolerate varying concentration of heavy metals, including copper, cadmium, chromium, lead, mercury, and zinc. This emphasized the resilience of microbial community in the face environmental contamination. Nutrient and contaminant level. Nitrate concentration exceeded previous record, indicating potential contamination. Total phosphorus level, especially in the urban areas such as Chobar, Subidhanagar, Teku, were notably high, likely attributed to the anthropogenic sources like septic system and fertilizer runoff. Coliform count. Elevated coliform count, particularly in Subidhanagar, Sankhamul, Teku, and Chobar, signify a potential risk of waterborne pathogen, highlighting the importance of the water treatment and sanitation measure in this area. Bacterial identification and antibiotic resistance. E. coli was identified as prevalent bacterium in the water sample, with all isolates showing resistance to ampicillin and erythromycin. However, susceptibility to chloramphenicol, nitrofurantoin, and genthamycin suggest potential alternative treatment option. The findings underscore the need for continued monitoring, effective water management strategies, and public awareness to safeguard the quality of this vital water resource in Kathmandu, Nepal. In this research, I would like to thank to my supervisor, respected Sir Professor Dr. Rajendra D. Joshi, for his continuous support, encouragement. And I would like to thank Mr. Pradeep Kumar Shah, my colleagues. He also support me during my research work. And Yogeshwari Mahavidyalaya, Ambe Jogai, BID, India, for providing us space and laboratory facilities throughout this research. And humanizing genomics, macrogene South Korea, for helping us to sequencing the gene of the E. coli. Thank you. Thank you very much, sir.
Yes, thank you, Pushpara Dayal. Pushpara Dayal is one of the senior faculty working at Nepal. The research area which he has performed is regarding environmental microbiology. He has taken one of the major problem in Kathmandu. Just like Ganga River in India, Bagmati River is main river in Nepal, which is carrying out environmental pollution and human health hazards. So he has taken the problem very keenly and studied on this problem very effectively and in deep sense. The physico-chemical analysis, the microbiological analysis, the isolation of indicators of pollution, Escherichia coli, which may be the indicators of fecal pollution resulting in typhoid, dysentery, cholera, and all other diseases are the center point of this work. He has also carried out some genetic analysis of the genomic studies of microorganism, and surprisingly, most of the Escherichia coli species, which is an indicator of water pollution, is found to have antibiotic resistance. Definitely, it is an alarming situation for the government of Nepal as well as the common people who are residing in the banks of the Bagmati River. Throughout the river, wherever it flows, the people who are residing there, they may have serious health in future. So the work which is carried out by Pushparaj Dal is very good. I congratulate him for selecting this topic for research. And after the research work is completed, I'm sure that this will be a draft of for the government of Nepal to work on it in future so as to eliminate the water pollution problem of Bagmati River in future permanently. So, congratulate Pushparaj Dayal for your continuous work. I hope that after PhD, you will continue your research in the benefit of Nepal people as well as the common society of Nepal. Thank you. Thank you very much.
Thank you. Thank you, sir. Thank you.
Thank you.
Okay, sir.
Okay.